Well, first of all, thank you very much for the invitation to come and talk here about characterization of carbohydrates, primarily dietary fiber, and also how it can be optimized in the development of the use of exogenous carbohydrates. The point which I'm going to address here today, they will deal with just a brief introduction, dietary fiber, nutritional effects, and also effects of exogenous enzymes. That will be rather brief. Then I'm going to talk about the diversity in dietary fiber in feedstuffs, and also how the fiber component can be determined with wet chemical methods, NIR technology, possibilities and limitations in this respect, and then a conclusion. If we are first looking into the nutritive values, and we have heard a lot of that during this meeting, I'll say I try to distinguish it a bit between what is the events that's occurring within the gastrointestinal tract and what is happening within the body. And as we just have heard in the previous presentation here and also yesterday, it's so that with amino acids there's a lot of things that's going on within the body. It's very important that you optimize the supply of individual nutrients relative to the demand for the synthesis and so on. I will say that for the with regards to the dietary fiber, although they in itself may influence also the uptake of nutrients to the body, it's also important how they act within the gastrointestinal tract in different perspectives, because most of their effects will be associated to that part of it. When we are looking on the enzymes that's produced by the birds, you'll see that among the different enzymes there are no fiber-producing enzymes. And it's also there, or no endogenous fiber-degrading enzyme, meaning that the more fiber, in general, the lower total tract digestibility of different nutrients. However, as also was explained yesterday by my peers, it's actually so that some kind of fibers, they may have an interactive effect on the gastrointestinal tract, meaning that although they can't be utilized by themselves, they may have a positive influence on the development of the gastrointestinal tract, and thereby it, you can say, potentially can influence the utilization of the nutrients. Just briefly about the possible effects of fibers in nutrition. Well, first of all, it is so that the cell walls, they are non-digestible by themselves. That means that whenever you have a cell structure coming from the feed that contains the fibers, this can in principle not be degraded by the endogenous enzymes. It's also so that some of the fibers, they may have influence on the viscous properties within the gastrointestinal tract, because in some cases it's so that the fibers that are situated within a cell wall matrix, they will, during the digestion processes, they will start to swell, and in connection with that process, some will leak out into the solution and thereby potentially influence the digestion of the nutrient, the rate of digestion, and also some of the absorption of it. Other kind of effects of the fibers in the cell walls, that is that they can encapsulate potentially available nutrients. That is particularly known, for instance, from some cell layers that's present in the cereals, for instance, the alveolar cell layers, which are very rich in essentially nutrients. They are surrounded by a cell wall that is very rigid and very difficult to degrade, thereby withholding the nutrients. The same is the case in many cases for cell walls coming from legumes. And you can see all the others are marked red. But in the other side of the spectrum, that is that the carbohydrates, the fiber components, they may have an impact on the act as prebiotic, thereby have a specific influence on the microbiota, and recently there has also been discussion about some symbiotic effects of the carbohydrates. The distinction between prebiotic and symbiotics, that is, that symbiotics apparently seems to act at much lower levels than is the case with the prebiotics. But what can be done with the exogenous enzyme? Well, first of all, you can say they can potentially hydrolyze and degrade the cell wall polysaccharides. It's also so, and that happens very efficiently actually, it can depolymerize and degrade soluble fiber, viscous fibers in the gastrointestinal tract. It's also so that they can act on the cell walls, encapsulating nutrient, and thereby also making part of that effects available. And then it's also so that they can help to produce prebiotics, symbiotics, and thereby influence the microbiota and potentially also the overall availability of the nutrient from a feed. To utilize that, however, it's required that you know the cell wall composition. You need to have methods so that you specifically can quantify the amount of nutrients that's present in the cell walls and also how they are composed. Okay, if we then are looking on the diversity in dietary fiber in feedstuffs, I can tell you, I can bring up this one, which is a cross-section, so you can see from wheat, and where you can see that there are fibers present in all these cell tissues from the outer part of the grain. You have the alveolar layer here, and then you have the cell walls in the endosperm. So they are all present there. And it's also so that but what is also very important, that is that these different cell layers, they have different composition. They have different composition within the grain. So when you have a composition of wheat, it's actually an average of what you have in the outer part of the grain, the alveolar layer, and also the endosperm part of it. In addition, going from one type of cereal to another, it's also becoming more complex because the cell walls of the protein-rich materials, they will look different to what you see here with the cereals. Then we can say, okay, the raw material situation for the poultry, they were more simple in the old days, and that may be the reason, partly at least, that they only required 15 or 20 pages for the first nutrient recommendation back in 44, and now they are more than 1,000 pages. Because when you're looking on how you essentially were feeding the birds here in the US, it was by using maize, corn, or whatever you call it, and also soybean meal. That was essentially the nutrient that we used. They are naturally variable, but not so extraordinary much. Then came the co-products of different sources. It could be DDGS that has been processed and which in itself has been exposed to different kind of liquids and enzymes before they are given to the birds. And then in addition, if you go a little bit more across the world, you'll also see that you have a lot of other raw materials that's available, and that is what you see in Europe. It's wheat, barley, rye, oats, co-products, soybean meals, not only soybean meal, it's rapeseed meal, cakes, sunflowers, and so on. Asia, a little bit more like you see in the US, and then when you come to Australia and also New Zealand, somewhat more like other kinds of raw materials. And they are naturally something that have an impact. And you can see the same when I try to one of the things that always good when you try to understand how the different fiber components, they act in the gastrointestinal tract. That is, go back and look on the fibers in the different cell wall matrix. What is their function within the plant materials? And for instance, if you're looking on the wheat here, you can see that the outer part of the grain, They are meant for protecting the grain against external invaders, as the fusarium or whatever you can have that can be invaders to the grain. Whereas the cell walls that's present in the endosperm, they have a very different structure to what you see in the outer part of the grain, primarily because they are surrounding the cell content. But if they have been as resistant as what you have in the outer part of the grain, this grain will never come to the point where it's germinated. This is with the whole grain. You also see some of the differences here when you start to process, and I've just taken an example here from where you have oats, barley, rye, and rice, and you then are looking on what is the concentration you have in the grain itself. And then when you are depleting, you're taking out the flours, you will see that you have much lower concentration in the flours, which are used for the human consumption, whereas when it comes to the co-products that's used in animal feeds, it is much more variable, it's much higher in concentration, and it typically concentrates also some of the more difficult degradable parts of the grain. Additionally, very much depending on the way they are processed, the concentration in the byproducts will vary far more than what is the case with the grains. The same happens to some extent when you come to the protein-rich crops and feedstuffs. One major difference is, however, you see, for instance, if you take soy, that is that the hull layer that surrounds the soy, they are not lignified, in contrast to what you see with the cereal grains. But then you can also say, in additional, it's so that they will be typically be extracted for the oil, which means that they are provided as feeds in form of meals and cakes, which means that they have also been through a processing step. You can have a look on some of these differences when you perform a enzymatic chemical gravimetric procedure for the individual sugar components that's present there. And what you can see, for instance, here, there is differences whether you are dealing with cereals up here, soybean and rapeseed meals down here. You'll see that in the cereals we have the higher concentration of arabinose and xylose representing the arabinoxylans. The barley has also high levels of non-cellulosic glucose representing beta-glucans, whereas you can see for the soybeans and the rapeseed meals, they are much higher in terms of uronic acids and also galactose, which are markers of the pectin-containing materials. You can draw a number of other, have a look on it from other angles as well. And that is also clearly shown when you perform a principal component analysis of the sugar composition of the non-cellulosic polysaccharides and also the cellulose content. Then when you are looking on how the cereals here, they deviate from what you see here with the protein-rich feedstuffs and crops. You'll see that they are primarily deviated here because of the uronic acid contents representing the pectin-containing materials. With regard to the lupins, there is further segregation because of the galactans. Whereas with regards to the cereals, they are all situated over here with higher levels of xylose and arabinose and some of the soluble glucose representing the arabinoxylans and the beta-glucans. There's also a segregation in the y-axis here because of some of the cellulose and the lignin content, which are higher with the sunflower and also the rapeseed meals as compared to the soybean meals and also the peas and fava beans. And you can also see for the cereals, the same goes here with lower levels with regards to maize and sorghum, and higher levels barley. Further, if you try to look specifically on the different cereals here, you'll see that the barley and the oats, they are segregated from the other cereals primarily because these two crops, they have a hull layer which surrounds it, whereas the hull layer with wheat and rye and so on, they will be lost during the threshing process. But when you're looking on hull-less barley and also hull-less oats, you'll see that they are more closely related to the other cereals, going from which are, however, separated here on an axis that goes from lower to higher levels of soluble arabinose islands in general. Okay, I'll see I have to hurry on a little bit. Well, just how can we determine it? You can say we have the chemical gravimetric procedure, which are the neutral determined fibers, acid determined fiber, acid determined lignin, and crude fiber methods, which are, you can say, some classical methodologies. There is the enzymatic gravimetric procedure, total, soluble, insoluble dietary fiber, all performed gravimetrically. And then you have the enzymatic chemical gravimetric procedures, which can separate it up into soluble, insoluble, non-cellulosic polysaccharides, cellulose, and the lignin. And then here we also have the NIR methods, which are something that is under development in this year. And just briefly with some of how to determine this, you can see essentially you remove starch and sugars from your samples, and then you perform acid hydrolysis and you separate the fiber components into cellulose, soluble and insoluble non-cellulosic polysaccharides, and also lignins. And if you will have a closer look on this aspect, I have gone through some of this procedure and also some of the other gravimetric procedure in this book chapter. Okay, going into the NIR analysis, what is that? Well, it is a discipline on the interaction between the light and the matter. Light can have different wavelengths in the electromagnetic spectrum. The infrared region, that is covering from 780 to 2,500 nanometer. What's happening is that when you expose them to the electromagnetic energy, you will have the molecule to vibrate, and they will also in a way that when they are when the same energy levels is used as what we have in the, or with the same frequency as in the molecules, you'll see that you will have emission of light. And then can you also say there is a number of other aspects here. They can considered as weak springs holding together two or more atoms, and they will vibrate naturally, but there is also so that they will be vibrate more energetically when they are exposed to the electromagnetic energy. And I've tried to illustrate some of it here. Whenever you have so that the energy of the electromagnetic field, they hit the same frequency as of the molecule, you have a peak response, whereas if it's not, you will not have a response. It's not so that, for instance, if you compare that for a spectrophotometrical measurement, that you will have it so nicely peaks, because you will actually see that they are a continuum of information that's spread on the different curves that's in these pictures. And it really requires a number of more deeper mathematics to extract out the information from these spectrums. And yeah, as I said, we need to do some relatively fancy match. There is also, well, there is a different technology involved here. The match behind it has developed quite a bit in recent years. It's, you can say, basically, if I should try to illustrate it, and I can see I'm running a little bit out of time, that is that what you perform up here, that is that you have the data. This is the fiber components you have here. You have this is the spectra. You perform a spectral analysis of the feed. You have the reference data available, and then you create a calibration model. That is straightforward. You can do that. Then you have a separate point of data you have collected in an independent database, and there you also you use the calibration model, and then you make a prediction which can be compared to what you have in your analytical models. And basically this is how it's working in different ways. But what I want to share with you, that is some results from a recent published paper, and where they have looked on NIR calibration with cereals and cereal co-products. They have been studying cereal and its co-products, and they have been looking on the relationship between the analytical values for them and also what you have been able to how they relate to your spectrum. And what you can see here, they actually look quite nice. However, I should say they have done here, that is that they have performed the calibration on the model, and then they have on the database -1, and then they have done a cross-model validation on the remaining ones. And when you are looking here on how it looks with the R squares, you will see that they are indeed very, very nice, relatively high. It's lower when you come to some of the components, and that's one of the limitations. Some of the components that's present in lower concentrations. And what you also can notice, that is that the R squares on the calibration, that's a little higher that we see when you have the cross-model validation of it. And the same goes for the standard error of calibration relative to the standard error of the prediction, that the standard error of calibration has lower values than what you have with the prediction. However, this is when you do a calibration and you use a cross-model validation. They all look very nice, but they are probably also making the picture a little nicer than what it is in the real world. We've been working with a little bit with that also. We've been working with a more heterogeneous type of feedstock materials. We work with cereals, cereal co-products, and protein and fiber-rich feedstuffs. And what we did here was that we took our total database, what we had on analytical data, and then we separated it into 75% of them, which are represented by the blue dots, which are the one on which we created the calibration. And then we used the 25% nicely spread within the whole area of it as a form of independent validation model on them. And you will notice here that when you compare what we found in our studies, you'll see the R squares, they look actually quite nice, relatively high. What you also can see, that is that when you perform the prediction, you will see that becomes lower. But here you should think of that this is on a form of independent as far as it can be. But what you particularly see the differences, that is when you come to the standard error of calibration relative to the standard error of prediction, you'll see that is noticeably higher. Because what you essentially are doing, that is that you are taking the spectrum of the independent database, and then you make a new prediction of them and compare to to the analytical values. And you will also notice here that when you're looking here, this is about the CVs, what you have. In particular, note here that it's somewhat higher for what you do. How does that, however, compare to when you do wet chemical analysis? Well, if you take the data here, which are the total for this broad database on the different feedstock models, again, you have for total, the CV was 70, it was 15 for the insoluble. And then you compare to what we have found in the literature. I should say this is not entirely the same data we have here. They are done on different matrices. But what you can compare here, that is, what you see on the prediction, you'll see that they are somewhat higher to what we can find in the analytical procedure. But if you look on the standard error of calibration to what we found with the standard here with the analytical procedures, it's not so terribly much higher. It should be standard error of calibration should be higher than on the chemical database, because you can never predict better than what you have in the other kinds of. Another thing which you also should notice here, that is that when you come to the soluble components, and so far I have not seen any NIR calibration on the soluble components. You are more challenged because basically with the methods we are using now, they are based on determination, the total components, and then also the insoluble, and then subtract the insoluble from the soluble. That's the way by which we calibrate the soluble components. And you can see even here with the wet chemical, it comes with some variability, and I presume it would be substantially much higher, and maybe the reason to why this haven't been published so far. Okay, possibilities. It is rapid determination of the content and composition of the dietary fraction of importance when formulating data. We have already large databases for cereal and cereal co-products and protein-rich. There are large databases for that, but I should say some of the limitation, that is that we have weaker calibration with dietary fiber components that's present in low concentration. We probably also need to work more with independent databases to see how it's really how it's really working, how robust will it be, and then also we can say it may not be quite as suitable for the soluble components because in the database we have, this is already a component that comes with a higher variability than is the case with the one we determine directly. And well, briefly, I have mentioned this here or during my presentation. It influences the nutrient utilization to a large extent, positive and negatively. We conventionally use wet chemical methods for the dietary fiber content and composition. We can also do reasonable good prediction with the NIRs in cereals and co-products. But when we come to the components present in low concentration, we are still challenged. I don't think that is the critical point here, but it's more critical that we have some limitations with soluble dietary fiber components. And with these words, thank you very much for your attention.